Thank you, Dr. Parker. Okay, so welcome everybody. We're going to go ahead and get started with our talks. So just a little bit about the structure of our talks. Our students presenting will give roughly a 15-minute talk in that range of what they've been presenting on. And then afterwards there will be time for questions. So if you have any questions that you come across as they go through their presentation, what did that slide mean? What on earth are those figures saying? Anything, that would be the time to ask them, okay? And in between times, we'll have a time to switch where you can get up and leave if you need to, or step outside for a bit, okay? So we're gonna go ahead and get started with our first presentation of the day, which is a special presentation for us because we have a special guest today. Ashley Rivera is one of our graduates from Wayland, and she's gone on to do wonderful things, and she's going to tell us some of the wonderful things that they've been doing at Texas Tech. So I'll go ahead and get your talk up here. For that too, reset. It's okay. Okay. All right. And you have the floor.
Thanks so much. Hi, everyone. I'm well. Okay. Hi, everyone. Thanks for welcoming me back. Like Dr. Thornhill said, I just graduated last year. I was in your shoes, sitting there, watching y'all, or watching just as you're watching me. Myself and also a fellow Wayland alum actually conducted this research at Texas Tech University Health Science Center, and I'll talk more about my program in general tonight at the banquet, but I'm just going to kind of give you a rough presentation of what we did over a course of, this is going to kind of mind-blowing, we did this over a period of 5 weeks, so this is all kind of jam-packed, so I'll kind of show you what we were able to do within those 5 weeks. But it was design and validation of a power sequencing assay for myofascial trigger point susceptibility genes. A whole lot of mouthful. But to begin with, myofascial trigger points are tender points in hard muscle that cause pain. So primarily caused by muscle overuse, trauma, and eccentric muscle contractions, and results in the disruption of normal function of the neuromuscular junction. So if you take an anatomy, you kind of somewhat know what I'm talking about. And also, we were given this project from our physical therapy program, which is just part of our School of Health Professions now. And they kind of brought it to us and said, is there a way to tie genetics to this pain that, physical therapy, with the kind of an issue that they've been dealing with lately. And we kind of took it under our wing to try and understand the genetics behind these TRPs, and just in order to prevent further cases or even eliminate existing cases. So our objectives were to design and develop a power sequencing assay that will determine if mutations in the opioids, TRP, serotonin, and osteopontin genes are distinguishable between these patients. And funny story about these genes really quick. These 4 genes, we kind of split, Taylor and I. Some of you already know Taylor. We split them up and they became our children. 2 of them were mine. But they kind of became our own babies, and we kind of developed them and took them along. Mine were better than Taylor's. It just happened that way. We were also trying to design power sequencing and Sanger sequencing primers for each single molecular nucleotide polymorphism, and assign a limited protection for each SNP for the assay, and also look at the sample extraction requirements. It's a lot because it's so molecular, and probably like mind-boggling right now, but I'll explain a little bit more. So for our genes of interest was the opioid delta receptor gene, which helps regulate calcium in the muscles. The calcitonin gene-related peptide promoter gene, which is along and aids with muscle contraction. The serotonin receptor gene that promotes or helps regulate the serotonin uptake, and also the osteopontin gene, which is upregulated with muscle damage. So those being the 4 genes of interest, we're looking at a specific part of this gene sequence, or each part of this gene that causes a mutation. So a mutation is some kind of abnormal functionality with the gene, which is we're questioning whether this mutation, present or not, is actually triggering or causing these trigger points to happen. But it all starts with PCR. If you've taken any kind of biology class, or more so specifically with Dr. Reinhardt, you kind of know already what polymerase chain reaction is, and that's the first and most commonly used amplification technique for DNA, for replicating DNA, and uses the cell's natural ability to already produce it and constantly replicate it. And it's just kind of making billions of copies of a DNA sequence by denaturation, just breaking apart those two double DNA, or the double stranded DNA, annealing. You select primers. And when I say we select primers, primers are the two things that attach on this DNA sequence that kind of bind to the specific target you're looking for and kind of amplifies just that one sequence. So you have to make your primers very specific to your gene sequence to amplify the one section you want to see. An extension of just extending and adding on new nucleotides to this expanding strand. And within hours, millions of copies of DNA are produced. This is just following PCR. We had to check with via gel whether or not our PCR was efficient or viable. And what we did was we ran each of these PCR reactions on a gel. And here you see an NTC, that's negative temper control. That just tells us whether or not there was contamination in our PCR reaction. If it was contaminated, there was no way we could move forward with the assay. So seeing here, any kind of ghost band or anything we question means unintended product. That means the readers on our primers were not specifically binding to that part of the DNA sequence we're looking for. So we went ahead and reran this osteocontin and found out it was just kind of layover of this serotonin here to this well, and it was just fine. So we did that multiple times just to see if our PCR reaction worked. Once PCR was done and finalized, we moved forward with pyrosequencing. That's just like a simple diagram of what pyrosequencing is. This is essentially the sequencing assay we're looking to design. It's entitled sequencing by synthesis because it takes advantage of the fact that every time DNA is produced or replicated, a phosphate's released. It takes advantage of that, and each time a phosphate's released, it becomes a cofactor with APS and is actually brought on with an enzyme, sulfurylase, to convert it to ATP. ATP is then used by luciferase to convert luciferin to oxyluciferin. That emits a light. And each time a nucleotide is added, and you see like a peak arises on a pyrogram. So every time a nucleotide is incorporated, you see a significant peak for that specific nucleotide. And following that, aberrase, just kind of a resetting enzyme to kind of just reset the whole thing and start the process all over again. So this is essentially what we're doing and what we were trying to do in development with our assay. These are pyrograms. You can't really see the numbers too well. What we're looking for with these pyrograms is a blue, kind of a flag, kind of a red flag, or red flags were completely, they weren't viable, something didn't pass, something failed. Yellow was questionable, so we were looking for this nice blue, kind of a blue reading, and a good percentage of saying what nucleotide was present. And I'll show you in the next section here. Serotonin, depending on what nucleotide was present at that specific loci or a specific part of this gene, whatever nucleotide is present tells you whether it's mutant, whether it's wild type. Here you can see, you can't really see the numbers too well, but 99% T indicates that there's a mutant at that specific area of this gene. Serotonin, we found a mutant, a homozygous mutant, meaning it was fully present of one nucleotide and not both. And I'll show you what a heterozygous means in a second. And wild type, just indicative of, you can see it's the complete opposite. It's 100% C rather than T. So just this is what we would look at when we analyze the results, determining for this mutation, for this patient sample, where they mutant, where they're wild type. CGRP, C is a heterozygous mutant. It's present at 50% C and 50% T. You can see that the peaks are at the same level, at the same amount, and if there was a double peak or a triple peak here, you can see, it just means that there's 3 of those consecutive nucleotides present. Here, them both being present has the same level, same amount, just meaning that this is a true heterozygous mutant present. Now, following our mutants, and we found our mutants, we moved on to Sanger sequencing. Now, this is kind of a backwards track. Sanger sequencing is another form of sequencing that just takes advantage, it's the gold standard, meaning that it's just something we always rely to, look backwards towards, and what we use to kind of confirm any other sequencing assays, because this is what's been used for many years now. Just incorporates dideoxynucleotides to terminate the chain of A, C, G, or T. These results are kind of different. You'll see it in a second. They're just represented by different colors. So whatever colors come off on this electrophoregram is what they're called, determines what is next in the sequence. So looking at our Sanger sequences, once we found out and kind of recognized that these mutations were happening in these certain patient samples, we sent them off to these Sanger sequence. And this just kind of confirms the fact that our pyrosequencing assay worked and it was valid. So here you see at loci about 154, the mutant, as I showed before, was T. It's showed by a red peak here. Again, these are just indicative of colors. The peak heights really don't matter. They're just kind of more of presence, absence, which nucleotide is present or not at this specific loci. Here you can see a C and a blue, representative of just the different color nucleotides. So you can see it's just very different from a pyrogram. And that doesn't include any values, but just a presence, absence type deal. And in here, for Sanger sequence, for the CGRP, remember it was a heterozygous mutant, had 50% C, 50% T. And here, or C and T. And here you can see that it has an overlapping peak. That's the way, and indicative of a heterozygous. Both nucleotides are present, so they just overlap one another. Not a 50-50%, but just apparent by the two colors present there. Now, following all that, remember we had 5 weeks, or 5 or so, about 6 weeks total, the 5 weeks actually doing an experiment. We had to perform blind studies. Now, we took 2 samples per gene. Taylor and I both kind of randomly told the other to look away or just left the room. And each of us picked a PCR product or a PCR patient sample and just said, okay, we'll test this and see what you get. It's kind of a blind study in that, depending on what percentage we obtain from the pyrosequencing assay, and that's the whole assay, from PCR to pyrosequencing, what results we were obtaining, we actually got 12 for 12. So he selected 6, I got all 6 right. I selected 6 for him, he got all his right. So we got, I guess, 100% correct on, I guess, our accuracy. And we were actually able to kind of designate what percentages were high and low, depending on how many, on the percentages you saw earlier, the pyrograms establish what was the low, what was the high, what was acceptable from the how many reruns. We actually did quite a few pyrosequencing runs, and just seeing those multiple samples, running only 24 samples on this, it's a 24-well plate that runs multiple samples, and just seeing the results of how many percentages, kind of gave us a range of what to say, what was acceptable, what was not. So we were able to conduct and say we had 100% accuracy. It was only 12 times, but we did validate and were able to kind of test each other in that way. And we also were able to establish a limit of detection. So whenever you're making this pyrosequencing and loading your PCR amplicon into the pyrosequencing reaction, you kind of can play around with how many microliters you're putting into this reaction. So we played around we started with about 15 and kept going down, progressively downwards until we reached 5. 5 was a limit where we can actually detect and get our blue readings that we wanted, and we're actually able to say there's mutant, there's heterozygous, or mutant, there was heterozygous where there was a wild type or normal sample present. And also just telling us with 10 nanograms of DNA, initially with the PCR reaction, which there's so many factors in the PCR, but whenever you're first loading DNA, you can kind of designate, based on your concentration of DNA from your patient sample that you extract, how much you're putting into the PCR reaction. So for assay results, opioid, which was Taylor's, not mine, ended up being a complete failure. So we could never sequence him correctly. Him, say him. They were children, they were him and her. They were actually, we were never able to sequence it all the way correctly. We were never able to get that nice blue reading. Even optimization of PCR was a little finicky and just wasn't working. But for CGRP, serotonin, and osteopontin, the SNPs, or the single nucleotide polymorphisms, those mutations that we're looking to see, are actually able to be sequenced 100% of the time. And also, again reiterating that 10 nanograms of DNA for the initial PCR reaction was viable, and only 5 microliters of that PCR amplicon can be used for pyrosequencing. For future studies, we gather a larger sample size to increase validity, more than the few that we actually ran, the 12 samples we had, and also determine a minimum DNA required for the initial PCR reaction. So play around with that first PCR setup, and also look at specimen collection, so blood versus buccal swab. The actual collections that we used were kind of just old DNA samples from past students. We're just kind of given DNA and said, run with it, see what you can do with it. But also looking at isolation requirements as far as PCI, which is the method of organic isolation, hygienic high-cube isolation. So this is all ways of extracting DNA from a sample. It also sequence patients to determine if there is a correlation between these mutations and the trigger point formation. I got really scared using the word correlation. But just seeing, like, if a patient has trigger point formations, do they also have that mutation? Does that make them more prone to it? And is it a warning for future reference? Just can this be tied on a molecular basis to whether or not the trigger point formations can happen? So I'd like to thank and acknowledge the following people. The first three, Dr. Troy Cooper, Phil Sizer, and Brad Allen, were just the professors that brought they're from the physical therapy program, that just brought the idea to my professor's attention. And Dr. Eric Hendricks, Kemi Carter, and Katie Bennett, all of my professors at TTUHSC that have put up with Taylor and I throughout the entire project, all the questions we asked and all the kind of mess-ups we would have. And also all of you for welcoming me back. That felt so great. And I kind of brought up the idea of Cornell's, hey, you know, Taylor and I both kind of questioning whether, what if we went back? The fact that us both being Wayland students and they paired us together just happened by chance. But we kind of were really hoping and getting for. She's coming back here and showing off what we've done. And I'll talk more about what we do at the banquet tonight. But thank y'all. We had a lot of fun.
So these single nucleotide polymorphisms were already known for this?
Yes, yes.
So you didn't,
So,
you didn't
no,
find
we didn't discover them, but we know they were present and they had been shown to be present in, didn't know that with that mutation, it was apparent that something was off. Like they have normal gene sequence for normal kind of muscle level, what was serotonin having normal functionality. That one specific SNP that they had researched beforehand and they noticed was kind of off with muscle dysfunction. Anything that related to muscle dysfunction or muscle, some kind of muscle abnormality was tied to these 4 SNPs. We were just kind of given these SNPs. We weren't really assigned, go find some. We were just kind of given, hey, these are questionable. Test these, see if they're related to the trigger point.
So you haven't looked at people that have the trigger point problem versus people that don't?
No, we weren't able to. We only were able to research the patients samples we had. That's kind of the next step is taking actual patients that we know have the trigger points and seeing if that's present or not. We're hoping it does, that could be a preventative measure and see, for screening and such purposes, but we couldn't really. That's one of the things we have on our long list of things to do. That was one of our last things. Can we take real patients that we know have it and test it? But that'd be the next step. We only have 5 or 6 weeks to actually do this, so that was one of our things to do it, but that's technically the outcome of what we would like to see.
That's one more question.
Mm-hmm.
Okay. So are there different kinds of drugs that people can take to help this problem, that the drug choice would be informed by which genes might be mutated? Like if serotonin screwed up, do you do some kind of serotonin blocker or?
Yes, depending on, depending on what the mutation caused, like if looking at, that'd be more like the in-depth study of the actual, the actual gene itself, not just, detection or not detection. Just think it's mutated or not, if that mutation, take that a step further. That's another question that I wish I could answer more so. But we didn't have that much of a time to actually look into it, but that would be, again, related to a future study, because we're looking at not only for detection, but what can be done, like preventative measures, like what drugs can we incorporate, and how would that affect the serotonin uptake, whether it stops it completely or promotes it even further, like whichever kind of methods it looks at. But we're looking at, that'd be a whole different kind of assay we have to design. This was just detecting patient or not. So I guess looking forward, that'd be a possible thing. But we weren't looking more at the drugs. We're kind of thinking, can we get it? Can we nail it and find it first?
Has Taylor gotten over his rabies?
I don't know. See, he's still kind of odd. He's still kind of a mad. I can say Yes.
Going along the line of the serotonin, so why did you pick serotonin? What does it do in the excitation action mechanism?
Like I said, we didn't get to choose what genes we could look at. These are just kind of based off of previous studies. whether or not we could kind of look for different genes that affected, but the serotonin receptor gene, actually it also has a role in Alzheimer's, different factors. We don't really scientists look up as far as what the effects of it would cause in the long run. I wish I'd known, like looking at it, just knowing that it's one of the most prevalent. I looked at it briefly. One of those that are kind of more prevalent in muscle, not damage. It's kind of like the overuse of the muscle, like too much serotonin, kind of causes this adverse effect and kind of promotes these trigger points. Trigger points can happen. So I mean, not looking too much into the serotonin itself, but like the research I did over it, that was one of my babies. It just affects the uptake of it. So either I would say it promotes it more. So it's kind of like the overaccumulation causes that really taut muscle and causes all that overproduction, like I said before. Like just the causing of it is just the overwork, the overuse of the muscle contraction, just the release of more serotonin, just cause that to kind of be more of the more prone to, and one of the genes that they decided to kind of give to us and to research. So and that's also kind of thought. This sounds really weird, but we're just kind of spoon fed the genes and stuff. So we kind of like adapt to what we can find with them to get around. One more.
So why the prior sequencing versus, is it
Singer?
Yes, yeah.
It's kind of more turnaround time. It's a very fast, like not saying too fast, but it's very compared to the PCR and such. It's just another method of sequencing that wouldn't take as long, that we could conduct with many more ranches. So I said 24 wells. You can actually test a whole lot of patient samples within those 24 wells. With Sanger sequence, it probably be within, depending on how many patients you have, it'd be kind of a mess of, like you saw the electrophoregram, it's just a whole mess, a whole mess. And with the Sanger, or with the Pyro, you can actually individualize each patient and see their sequence, their specific sequence within each well or within each reaction that you see. Because you can perform each well to have a different sequencing primer and attach to a different part of the look site. So you can technically test different patients at once and still see them apart versus Sanger that sequences all in one all in one pool, and you can't really pinpoint necessarily which patients which or determine which mutations where, just because all the patients are pulled together. So it's kind of just a better method of individualizing patient results and just improving turnaround time. Turnaround time meaning just making it faster, produce faster results. And
What about cost?
cost. It is a little expensive. That's one of the downfalls of the pyrosequencing. I mean, a pyrosequencing we have in the lab, not every lab does. So, I mean, just the cost-wise, it'd be for more developed labs, but with the way this field's moving forward, I mean, it's bound to, lots of these issues are bound to be in every lab. So.
Pick your favorite slide and stand in front of the spigot over there. This is for everybody giving the presentation. Okay.
Well, I know.
10 seconds, go. All right.
I have one more question. My question is, in the pyrosequencing, so you have different height of your
Different heights,
spikes,
yes.
right?
Yes.
So can you explain how is that possible for the height differentiate those among those nucleotides?
Like why is the height different?
Yeah.
Or like, okay, why, well, it's just, like I said, it's based on the amount of pyrophosphate that are released. So, the more that they are released, just means like it's this here, like, I can't really see what letter it is, let me see here, G. But, see how it has like the triple of that height, or that peak, it just means there'd be, if you were reading that sequence, it'd be GGG. I can't see what letter that is, but GGG versus just being 1T. This would be kind of a 1C or T, whatever. Can't really see, but one nucleotide versus maybe 3 here. So every time we see a peak height difference, it's just kind of another fold. So this would represent one nucleotide. Here would represent another 1, there's a 2, there's a 3. So whatever nucleotide this is, just incorporates when you're reading a sequence and you're writing out the sequence, be 3 in a row versus just 1. So whenever you're writing out the sequence, it would be like the electrophoregram that has them all lined up, nice and pretty, on top. more than one nucleotide present. Does that make sense?
Well, let's thank our speaker again.